Good afternoon. My name is Mark Nardone. I'm director and co-founder of Bio-Trac. Welcome to our Thursday weekly webinar series. I ask that questions be submitted in the Q&A box, and they'll be addressed at the end of the presentation as time permits. For those who aren't familiar, Bio-Trac offers hands-on training workshops for research scientists. These workshops, which are team taught by active researchers, focus on the latest relevant techniques that are necessary for laboratory research. Originally based at the National Institutes of Health for 30 years, Bio-Trac is now located on Montgomery College Germantown campus in the Bioscience Education Center, and it's open to the scientific community. Since the program's inception, we have provided hands-on training workshops to over 17,000 scientists. Along with our hands-on training workshops, we continue to serve the scientific community by offering bio panels, custom design training workshops, weekly webinar series, hosting government and industry retreats and symposiums, as well as other key scientific events and outreach initiatives, such as the Maryland Science Olympiad. Today, it's my pleasure to introduce to you Dr. Elke Bergmann Leitner. Dr. Bergmann Leitner is the chief of the Flow Cytometry Center for the Malaria Vaccine Branch at the Walter Reed Army Institute of Research in Silver Spring, Maryland. She is an adjunct research associate professor of medicine at the Uniformed Services University of Health Sciences and has been teaching flow cytometry for over 25 years. Currently, her main research focuses on the development of readout methods capable of identifying immune correlates of protection against infectious diseases. As director of Bio-Trac, I've had the pleasure of working with Elke for over 20 years, as she is the workshop director and key contributor to our triannual hands-on flow cytometry training workshop. This workshop has provided training to over 1,000 research scientists from local, national, and international institutions. So without further pause, I'd like to welcome Dr. Elke Bergmann Leitner in her presentation on rare cell detection by flow cytometry. Elke, they're all yours.
Thank you very much, Mark, for the introduction. Let me say that I've thoroughly enjoyed the many years teaming up with you for flow cytometry, immunology, and the various workshops. And I'm proud to be part of such a high quality program. So for today, you heard that I'm teaching normally the flow cytometry, and one of the things that are very of big concern to me is when people work with very small sample sizes, or they're looking for populations that are fairly rare. And so we addressed that experimentally over the years. And today I wanted to give you two examples with all the scientific and methodological background around it, how you can actually make this work. Before we get started, Mark already kindly introduced me. So I'm at Walter Reed, which is the premier research institute for the Department of Defense. In my core unit, we are evaluating immune responses to vaccines, to diseases. Primarily our focus is on malaria, but we've been involved in other mission critical vaccines, including the Ebola vaccine and now Operation Warp Speed, so the COVID vaccine. So what I prescribed to a long time ago is that in order to find a correlate of protection or a correlate of disease, because it can go both directions, it is rarely enough just to have a handful of measurements. So we know the classic interferon gamma IL-2 TNF CD4 versus CD8 panel, but rarely has that shown to point out for viral diseases or for parasitic diseases like malaria that this cause protection. It is just a measure of immunogenicity, and that is okay in its own right. But to really find out what makes a vaccine work, or what makes a vaccine component like the adjuvant work, you really have to have a large number of measurements. And that's what we call immunoprofiling. You will also hear people talking about establishing an atlas of landscapes of various immune responses. Also would like to say that while we have a focus here on immunology, the rare cell detection that I'm describing, you could apply to any other disease model. So the first thing that comes to my mind is when you look at tumor infiltrating lymphocytes in a biopsy from a cancer patient. So while my examples are immunologic, they can be applied to other diseases. All right, so what we do when we do the profiling, we collect a wide range of immune measures. So it's anywhere between 40 to 280. In one case, we had over 1,000 immune measures and features. And then we do your computational integration, then univariate, multivariate analysis, and then we do a lot of modeling. And this will then give a prediction of what are the markers or what is the marker that distinguishes between success and failure, if you want. And in one case, we're even able to go back and address that in an experimental setting. And we're able actually to validate what we had found through modeling. So one of the main tools for this profiling, of course, is flow cytometry. Otherwise, I wouldn't be talking to you about this. And so just as a quick overview, flow cytometry can be used as a tool for many different applications. Now for immunoprofiling, we use it to identify cellular subsets using lineage-specific markers, analysis of specific molecules either on the cell or in the cell. You can think here of cell surface markers, homing receptors, other lineage-specific markers, factors that are being produced because the cell is activated or you activate it in vitro, and you can measure these factors intracellularly. Monitoring the activation differentiation of cells, that would give you an idea, what is going on in the body. In pathology, flow is used to detect infections using antibodies that would detect pathogen-derived antigens. Same thing applies to tumor cells. If you have tumor-specific antibodies, you can measure or you can detect tumor cells, as I said, in a biopsy, for example. You can monitor chronic diseases and the cellular events going on, presence of specific DNA sequences in certain cells, because we now do a lot of the prime flow where you have antibodies, where you don't necessarily have antibodies, but you have molecular probes that are then fluorochrome labeled and you get hybridization. Cell cycle analysis, not to forget, that help you to monitor activation events, or when we go into the tumor field, where you could actually test biopsies and whether or not tumor cells in there will respond to a chemotherapeutic drug by stalling in their cell cycle and so on. Now, more and more are we going towards high-dimensional flow cytometry, also called cytomics. And what that really entails is that you have many, many different markers because you're trying to drill in and get an in-depth analysis to get a lot of granularity. Regular flow, in my mind, up to 6 colors would be regular flow, where unless you have a very rare cell type already here, you can overcome this by acquiring more cells. We typically use it when we do phenotyping of blood cells, or we do functional assays, so whatever that may be, whether that are killing assays or phagocytic assays. And again, sample size can be an issue if you have, if you study populations that have a fairly low frequency. Now, even if you have a population that's about a 1% frequency, if you are further characterizing that population where you have then different subsets, you are running into an issue. And that's typically what we have with the high dimensional flow cytometry. And in the end, when you do the analysis, you will need a specialized software and ideally a bioinformatics person that can help you with reducing the dimensionality do the cluster analysis, and then do either t-SNE or SPADE. Now, if I go further into the advantages of the high-dimensional flow cytometry, apart from what we already said, where you can get high granularity and you can really define changes in the population as it regards to their activation, their functional activity, and so on. If you have a sorter associated, or your flow cytometer is a sorter, you can use it as a preparative tool for cell isolation for downstream applications such as RNA-seq. And early on, when transcriptomics became very hot, people would take, at least here for me, for vaccine applications, people will take the blood, the peripheral blood from vaccinees or even from patients, and they would run transcriptomics. And same thing, there's a lot of noise and a lot of cells are not involved. And so everything kind of stays in the murkiness. And the data that are coming out now that people are sorting specific cell population, they actually reveal finally differences, and they shed light, or they uncover things that we have been looking for for the longest. Now, what are the disadvantages? You really have to optimize your flow panel before the actual experiments. If you have an instrument that allows you to do ideally 50 different markers, that doesn't mean you will get it to work. And it really depends how you set up your panel. And because not all flow chromes play nicely with each other, and the more colors you put in, the more potential bleed over and interference you can get. And so that means you have to get a good flow panel. You have to work on this. It will be heavy on assay controls. And then once you set this into action and you start acquiring your data, a lot of times it will require you have a specialized software that's easy to handle, where the audit is easy. I'm thinking of one of the softwares that pretty much most people use, but it's a nightmare to audit. And so that is one thing to keep in mind. But anyway, so specialized software for data mining. Gating strategy are very crucial, and that you have already prepared a gating strategy. And you always follow this down to the T, that you follow the same sequence of gating strategies. Also, will you do manual gating versus automated gating? Will you do all this analysis of your high dimensional flow data? Will you do it manually, or will you actually have automated analysis? These are all things, that will impact the quality of data of your results. And I'm getting at today's webinar is the sample size calculations, and to make sure that you acquire enough cells to have statistical power, and also that you keep an eye on your frequency of your cells, because we know typically the threshold of detection is 1 in 10,000 cells. Now, so how do we address some of these issues when we talk about the frequency of the cell types that you want to analyze and define? Sample size calculations. That, of course, means that you have to run initial experiments, bait cutting experiments if you want, where you find out what is the frequency of your cells, and then calculate up how many cells you have to acquire. Is it enough to have 1 million, or will it require 20 million? And this is not out of the question, so I'm not exaggerating with these numbers. So the further you drill down, the more markers you have, and the more diverse every cluster becomes, the more you have to be aware of that you need to have your sample size correct. Then acquire based on lineage-specific marker as a trigger, that you're basically saying, all right, I only gonna acquire a certain main population, or I use this as a trigger. So if I say I'm acquiring 1 million cells, it should be based on this particular marker. Or it could be an activation marker in case of immunological assessments. How to increase the signal-to-noise ratio, coming back to the flow panels. And then, and this is what today is all about, is magnetic enrichment based on specific markers. In my case, it would be activation marker. If you work in the cancer immunology, maybe it would be tumor antigen. Or it could even be some kind of other marker that points out the cells that you need to study. Now, when we now go down the road of immunological analysis, the frequency of antigen-specific cells can vary quite a bit. And so a couple of years ago, Petra Bacher published this, where you can see how variable this can be. For example, naive T cells, you know, the older an individual gets, the less of naive cells you will have. With vaccines, and that's what we're working on, it really depends on the potency of the vaccine and also whether you already have pre-existing immunity. If you have, for example, influenza vaccine, well, you already had influenza-specific T cells in there. And so your frequency is definitely higher as if somebody gets a vaccine for Ebola, where you have your the precursors from the T cell receptor rearrangement, but that's about it. And so here for standard flow cytometry, it's accepted that a frequency of 1 in 1,000 to 1 in 10,000 is pretty much the range of what standard flow cytometry can pick up. If you're going if you're going beyond this, so if you're going lower than 1 in 10,000 cells, then either you have to come up with some form of enrichment, and then you can actually increase your signal, amplify your signal so we see it. And I will show you the data and also the proof that you're not really now studying an artifact. So the two examples I will have are both for the analysis of antigen-specific T cells and their function. The first one will be where we use a cell surface staining to differentiate different subsets of follicular T helper cells. Here, you know, for those of you that are not so familiar with T cell immunology, there are different subsets of T cells as they can mature and differentiate. And so when we do this characterization and we do enrichment, now within that subset of Th1, Th2, and Th17, so these three, if you take them, now if I enrich, do I enrich them equally? Or do I now change the composition of the overall population of the overall follicular T helper cells? And with that, now I'm following a red herring. So that will be the first example. The second example will be the functional assessment of antigen-specific cells based on cytokine production. And that is actually something that we is right now has been submitted as a manuscript. So in the past, you know, when you go to the various websites where they sell magnetic bead labeled antibodies, they're normally shy saying that you could use it on fixed and permeabilized cells because that in itself can cause a lot of non-specific background. And I will show you the data that you can actually get it to work. All right. So when I first show you the data on the 154, so that will be our first activation marker, and I'll also show you then CD69. So both are activation markers expressed by T cells as a result of antigen-specific stimulation. This has been accepted as a marker of antigen specificity. Now here would be our control stimulation of PBMCs, just with heat-stable antigen. That is our choice. And here the contour plot shows us CD154 staining versus CD4 staining. I don't show you upstream gating where, you know, the classic one, forward sideward scatters, singlets, viable, and then CD3. I'm just showing you, you know, the final gating. The gate is set with fluorescence -1 controls. So that would be a control stimulation, tells you, all right, this is our background here, 0.3%. Now, if we have a specifically stimulated aliquot, then this is actually fairly high, and there's actually a virus-specific response. So we get from 0.3 to 1.6. So we would have about 1.3% of our CD4 positive T cells are antigen specific. That's a very high frequency. Now, if we do the enrichment based on 154, so you would have a magnetic bead labeled antibody for 154, and then through a magnetic column, you are enriching. We can bring this frequency up from 1.6 to about 48%. Or if we, yeah, about 48%. You may say, well, shouldn't I get 90 or 100%? No, because other cells, such as CD8 positive cells, also can express 154 as a result of stimulation. So here we are just looking at CD4 positive cells. Okay. Now, what is different about our approach, or what is new? We can actually now, oops, I'm very sorry. We can, by having, instead of 1.5% of cells and then drill into the detail of that population, we are starting out with about 50% in that particular example. And with that, you can increase the frequency quite dramatically. And with that, you have a better sample size. Now, when it comes to the magnetic enrichment of fixed permeabilized cells, so cells that have been prepared for intracellular staining, so far has been deemed unfeasible. And I said, I will show you this. And so I would say, let's jump into the first example. So as a very brief introduction, follicular T helper cells, they're important for driving antibody responses. They're normally found in B cell compartments of lymphoid tissues, but the circulating follicular T helper cells, they're actually found in the blood. And they are defined by, this is a flow panel that was published by the Ueno laboratory. So where you can differentiate, you can distinguish between TFH1, TFH2, and TFH17 based on the co-expression of CXCR5, of course, with either CXCR3 or CCR6. TFH17, TFH2 in vitro are good B cell helpers, provide good B cell help. TFH1 in vitro not, but in circulation. So when you take blood from somebody that had been recently vaccinated, at least for influenza, this also correlates with antibody production. So there's a little bit of this discordance between in vitro and in vivo. So how would you go about this? So here would be our flow chart, the logistics of setting up the assay. So first of all, you stimulate your cells with specific antigens versus control. Then you incubate overnight. And then next day, you come in and you label your cells with duochrome and magnetic bead labeled antibodies. And now you can, and that's what we've did for quite some time for QC. You can take an aliquot right then and there and take it into a flow cytometer. Or the main thrust, the main proportion goes into magnetic bead enrichment. You can do this manually or automated, put it over the magnet, and then acquire it in a flow cytometer. Why I say manual versus automated? Automated, there's one flow cytometer that has already a magnetic column incorporated. So everything is hands-off. You just basically take the sample, you stain it with a cocktail, and then the instrument will do the magnetic bead enrichment, the elution, and then the flow cytometric acquisition as well. So the gating strategy here for the rare cell detection is, again, CD3 positive cells, you gate then for CD4s, then based on the 154 expression, so all the antigen-specific CD4 positive cells that are then gated. Again, the gates are set with FMO controls. Then we look for the expression of CXCR5, so these are follicular T helper cells, and then we are drilling in, and this is why we need a good sample size, and really amplify the signal out of the noise, because now we want to go ahead and based on the expression of CXCR3 and CCR6, now we can assess the frequency of these different subsets. So that's the gating strategy. Now, if I don't do this and I run it unenriched, that is this data, you can see here we are there, and this is for the Ebola vaccine. We had 3 different cohorts with increasing vaccine dose. It is a single vaccination on day 0, so that would be your baseline. Day 28 after vaccination and day 56 after vaccination. Here the line I put in is basically the accepted threshold. So you can see there's nothing to report. It appears that the data would tell us if the vaccine does not induce specific follicular T helper cells, or it could be that the signal-noise ratio is too high, and therefore we cannot measure these cells. Now, that other aliquot from that culture was magnetically enriched, and that's the data that you see here. Same thing, but now this time we have in what we put in the flow cytometer the 154 enriched cells, as you saw in the previous flow chart. And now you can see that in all 3 vaccine cohorts, do you see a significant induction of specific follicular T helper cells. With that, we would conclude the vaccine was successful. And it's not just simply a higher noise, because our D0 responses, our baseline, is still down, and we have significant changes. Now, as we drill down, so we are still on the TFH. Now, as we drill down into the subset TFH 1, 2, and 17, maybe that's where we get into the artifacts. And for this, trust me, the reviewers asked that for this figure. When you compare overall, the overall frequency, so when you go back to the grandparent population, when you compare the frequencies of all the follicular T helper cells that are specific, so 154 positive, you can see there's no difference. The box plots are fully overlapping. Now, when I drill down into the subsets, same thing. I do not see a statistically significant background difference. Sorry. So from that, we can conclude enrichment does not alter the absolute frequency of follicular T helper cells, and the absolute frequency as well the composition of our CTFH subsets. So for the summary for the cell surface, for our first example, we can say it worked and amplified the signal, allowed us to pick out these rare cells and study them further. It also gave us data that we could then use for our data integration, and turned out that these CTFH17 cells were positive correlated with the persistence of neutralizing antibody titers to the Ebola. All right, so second example, I'm seeing that I'm slightly running behind time. Sorry for that. So the second example is that we are now preparing the cells for intracellular staining. So we are starting out with the same thing. We stimulate cells with specific antigen versus control. 2 hours later, because we want to measure cytokines, we block the secretion of the cytokine from the cells with a Golgi block. Next morning, we harvest the cells. We prepare them for cell surface staining with our antibody cocktail. And then after the stain, we prepare the cells for intracellular staining. So we fix the cells, we permeabilize them, and then we proceed with the intracellular staining, again with a cocktail of antibodies. And very similar to first example, we take an aliquot, so where we can see what was our starting point and how the frequencies change. And the rest of, or the other aliquot goes into enrichment, again, either manual or automated, and then acquisition in the flow cytometer. Again, you don't have to use this particular flow cytometer. We've worked with other manufacturers. We have other instruments in the lab, so that is not specific for a given flow cytometer. I want to point this out. And make sure that this is not something where you think, oh, I don't have that flow cytometer, I can't do it. As long as you can do the manual enrichment, the flow cytometers make so much of a difference in most cases. All right, so what is our gating strategy or our analysis? So we first gate for the lymphocytes, then for the singles, then we gate for viable CD3 positive cells. Then you can gate for either CD8 or CD4 or CD8. And here, we just gated for this example, we gate on CD4 positive cells. And then we said, all right, within this gate here, how many are positive for interferon gamma? So again, the gates are set with FMOs, so where you know where the cutoff is. Now, first of all, the ICS preparation did not impact the enrichment efficiency. So what you see here is when we initially did just polyclonal stimulation, because if that doesn't work, then we don't have to go any further. So here would be the unenriched versus the enriched. And when we look at the overall frequencies, we don't see a statistically significant difference whether we looked at viable cells or fixed permeabilized cells. So that did not change. When you do the enrichment on viable cells versus fixed permeabilized cells, for 154 in this case here, again, no statistical difference. So it's not that you are through the ICS treatment or preparation that you are now, you get so much stickiness that everything starts to stick and everything is positive. And that's true for the nasal stimulation as well. Now, if we go ahead and we look at other stimulations which are with a much lower frequency of the cells, because polyclonal is easy, as I said, we see the same thing, that first of all, we don't increase on our non-specific background, but that with enrichment, you can increase the overall pool or the frequency of your antigen-specific cells here for CEF. So that's a pool of class 2 peptides from common human pathogens, tetanus toxoid. Again, this runs as a positive control and background. And here we compared manual versus automated enrichment. And you see that in case of intracellular staining, the auto enrichment is more efficient in some in these cases. So it works slightly better, but in the end, if you don't have this particular instrument, you can do it with manual enrichment. Here we looked at interferon gamma producing cells, and same thing, we can get a drastic increase in the number of positive cells by enriching. Let me see what I have here. Oh, I should wrap up very soon. Hopefully you will see this soon in print. This doesn't just work with 154, but also with CD69, which would be more universal for all T cells. 154 is all CD4 positive cells and a subset of CD8s. CD69 is for T cells as well as for B cells. And so if I just summarize here, enrichment gives you a significant increase in your sample size. At the same time, though, it does not skew your overall composition if you look for subsets. I want to finish up here with a power analysis comparing the effect, size, and variability. So what we're trying to do here is show statistically what enrichment can do. So here we calculated the minimum detector fold change of a reference sample to your experimental. And then we ran 10 versus 15 versus 25 donors that were stimulated always with the same thing, and then run enriched versus unenriched. And so we are reporting here for two different statistical tests, either two-sample t-test or paired t-test. We are reporting that the fold change as a factor of, or as a function of variability here as the coefficient of variation. And based on what we typically observe, the CVs, they were observed with enriched samples versus unenriched samples. This is what we shaded. And so what you can see is that with enrichment overall, you have a much lower percentage CV because you have a stronger signal, right? We know that from many other systems that the higher you signal, the lower the variability. And the other way around, the closer you are to your detection limit, the more your variability you have. So with that, those statistics show you that enrichment can dramatically, statistically significantly reduce the variability that you have, even if you have a smaller number of donors, right? Okay, so just to finish up, enrichment based on either 154 or CD69 does not skew composition of T-cell subsets. Enrichment of fixed permeabilized samples does not increase non-specific signal. So it's not that all of a sudden you get some nonsense data. The enrichment of antigen-specific cells enables you high dimensional flow cytometry because as it increases the sample size, it reduces the interassay variability and even intra-assay variability, and then increases your signal-to-noise ratio. And with that, you get data sets that have sufficient statistical power to avoid the will avoid that the identification of the immune correlate is not reproducible. Because often you find certain labs where report, oh, we found the immune correlate. And then next study with the same vaccine, oh no, our immune correlate has slightly changed. And in reality, it's just, it's most likely something technical, or when you do this computational integration in the modeling that you overfit your model. All right, need to finish up. I have to thank 5 people in particular. So all the work around Ebola was done by soon-to-be Dr. Farouk. The work on the enrichment of ICS-prepared samples were done by Dr. also Lieutenant Colonel Marie Morat. She's a postdoctoral fellow in my lab. I'm very lucky to have her, as well as the other people such as Ms. Duncan, very valuable and very, very highly motivated individual. And my 2 long-term collaborators, Dr. Kevin Beck, who's now with Antigen. I couldn't have done any with that without him and some of his vision, and Dr. Chaudhry, or now Captain Chaudhry, for all the computational support. With that, I close. I'm very sorry that I'm 5 minutes over time, and I'm taking questions.
All right, Elke, thank you so much for a wonderful presentation. Going into the questions, first one, what would be a sign that 1 million cells would be too few? And the follow-up, what would be indicators of needing 10 million versus 20 million cells?
Sample size calculation. So you would run one experiment where you're doing the staining that you would, that is your experimental panel, and you determine what is the frequency of the cells that you're interested in. And if this is either the parent population, or if you want to further break it out, then you would do sample size calculation. Statistical programs can do this. They will then tell you how many cells you have to acquire. So it would be statistical tools.
All right, next question. For this type of enrichment methods, do you use frozen PBMC?
Yes, it works with both. It works with frozen. So almost exclusively there. I had to pause because we mixed some fresh samples into the power analysis, but we've done it with both fresh and fixed. And the majority, the thrust of the work was done with frozen PBMCs. Some of them actually were frozen for, in one case, 10 years.
Next, does using magnetic beads differ for intracellular staining compared to surface marker staining?
No, it doesn't. So the important thing is that you titrate the antibody fairly well, and then it does not it does not make a difference.
And last question, do you think using beads may block the marker of interest in analysis later on?
Um, not in our hands with 154 and 69, but if you are concerned, there are reagents available where you can clip off the magnetic bead and then either come back with the same antibody flow count label, where you then run into the issue about epitope occupancy, or you just come back with a secondary. But there are reagents available if you're concerned where you can clip off the magnetic bead. Just keep in mind the magnetic beads that we are using from the one company is about 40 nanometers, so it's about virus size.
Very good. There was one question regarding accessing a preprint of Mura MJ Zatel's just for the study of figures and procedures. Is that available at this point?
Not yet. However, if they were to send you an email, me an email, as soon as that's the case, I will talk to Colonel Murat and see whether we can just send out the protocol.
Very good. Dr. Kaiser, email me directly. You have my email on the correspondences that have been going out. I'd like to thank everyone for attending today. Next Tuesday, you'll receive further information regarding the recording of today's event and Dr. Bergmann Leitner's contact information, as well as a PDF of today's presentation. Next week's webinar, given next Thursday, will be given by Dr. Richard Lee, who's from Johns Hopkins University. Richard will be presenting on bisulfite pyrosequencing, quantitative approach for assessing DNA methylation. And I hope to see everyone at next week's presentation. Enjoy the day and be safe. Thank you.
Bye.